Just wait a minute. So we did up to this. Okay. Classification of amino acid. Okay. Anushka, do you have any doubt from the previous class?
No, sir.
No. So we did classification of amino acid on the basis of the relative position. Now we are going to do the classification. Second classification is basically classification of again amino acids on the basis of pH value. pH value. Now, as you can see, this amino, this is NH3, basically NH2R and NH2amine, and we are talking about the carboxylic acid. Both are weak acid and weak base. So here, how we can classify this? If number of NH2 group is equal to number of C double bond OH, then the solution, or it is basically neutral amino acid. Neutral amino acid. That means basically the pH will be equal to 7. pH will be equal to 7. Now the second is basically if number of NH2 is greater than number of COOH, carboxylic acid, then since basic is more in number than acidic, so it will be basic amino acid. It's very easy, right? Basic amino acids, and its pH value will be equal greater than 7. pH value greater than 7. Then come to the next part. If number of NH2 lesser than number of COOH, then it is acidic amino acid. Acidic amino acid, and since it is acidic in nature, so the pH will be lesser than 7. It's very easy, right?
Done, sir.
Very good. Now the next Alan, Sakina, everyone done with this?
Yes, sir.
Okay, thank you. Now the next is classification on the basis of source. On the basis of source. I have sent two sample paper in group. Are you added in that? Anushka, Sakina.
Yes, sir.
So have you got the sample paper?
Yes, I got.
Okay, please do solve that before your board exams. Okay, it's very important.
Yes, sir.
So these are of two types. Sakina, what about you? And Alan?
Yes, sir.
So,
Okay, thank you. So on the basis of source, it is of two types, essential amino acids, and another is non-essential amino acid. Non-essential amino acids. Essential amino acid and non-essential amino acid. So basically, essential amino acids which cannot be synthesized in our body, in the body. So we can define it like the amino acid which can't be synthesized, which can't be synthesized in the body and must be supplied through diet. Must be supplied through diet. This type of amino acid is known as basically essential amino acid. And the examples are very important because in MCQs, it will be directly questioned that which one of the following is essential or non-essential amino acid. So example of essential amino acid is, here I'm going to write leucine, then comes lysine, then comes methionine. And what is non-essential amino acids? The amino acid which is synthesized in our body, which can synthesized in our body, that is already present. So we can write which can be synthesized in body, as for example cysteine, glutamate, glutamine. So please do write it. We can write also non-essential. Glycine is also non-essential. We can write also alanine is also non-essential, and we can write cysteine. I already written it. I'm going to write more example in this also.
Sir,
Yes,
sir, what's the first example in non-essential amino acids?
it is cysteine, cysteine.
Yes.
No, if you still didn't get it, please ask.
sir, done.
Done, everyone?
So,
Sakina? Yeah, Alan. Thank you.
Yes, sir. Done.
Okay, there are more examples that I wanted to write that here. VIP. It's just to remember it. V is for valine. Valine is also this essential, and then I is for isoleucine, and then this P is for phenylalanine. Sorry, phenylalanine. Then we can write PMT. PMT is stand for. This is all essential. This part. Okay. P is for basically same phenylalanine. M is for methionine that I have already written, methionine, and T is for tryptophan. So the example is very, very important. Let me know when you are done with this.
Yes, sir, done.
Very good. Now see, the structure of alpha amino acids. The structure of alpha amino acids. First of all, I would like to write the structure of alpha amino acids. So you know this is your carboxylic acid, and this is your alpha carbon to which NH2 is connected. So it is alpha amino acid, and you know this will be H, and there will be R or H, whatever it can be. Okay. If this R is, if this R is basically replaced by hydrogen, if this R is replaced by hydrogen, and you can see I'm going to write this C, and this R is replaced by hydrogen, and the rest part is same as it is, and the rest part is as it is. So you can see this H, this H. So its name is basically, this is your glycine. You can easily draw the structure of alpha amino acid, then replace this R with H, then it will be glycine. And if you are going to replace this R with CH3, then it will be alanine. So this is your alpha amino acid H2N, and then here it is C double bond O, and here it is C double bond OH. So as you can see, this is your CH3. So this is your alanine. So it just trick to remember it, and at least you should remember 2 to 5 alpha amino acid. Okay, so please do write it. Let me know when you are done with this.
Done, sir.
Done, everyone? Okay. Now there are some points that you need to write. Write with me. Alpha amino acids are optically active except glycine. Except glycine. Next point is glycine is sweet in taste. Glycine is sweet in taste. Next point, it is about tyrosine. Tyrosine is obtained from cheese. This tyro means tyros means basically cheese. So tyrosine is obtained from cheese. Please do write up to this. Then we are going to start the next topic, which is protein. Please do write it.
Done, sir.
Very good. So let's start protein. It's a totally theoretical chapter except that carbohydrates and very easy chapter. You just need to remember points in this chapter. Protein. See, protein. Here we need to study about peptide linkage, which is also known as amide bond, which is also known as amide bond. And how does peptide linkage looks? So it is like this. So first I'm going to give the definition of this, and then we will see an example. So when 2 alpha amino acids combine together to form, write with me please, peptide. By the elimination of water. The bond CONH, the bond CONH present in peptide is known as peptide bond. Is known as peptide bond. So basically, when you are going to mix or combine 2 alpha amino acids, there will be removal of water, and the bond between CO, as you can see, this is your CO part, this is your CO part, and this is your N, CON. So this part is basically known as CON, bond is known as peptide linkage. Is that clear? I'll give an example. With the help of a reaction, you will get. First, please do write it.
Sir, done.
Very good. Sakina and Alan.
Yes, sir. Done.
Done. Now I'm going to take an example. As you can see that there is an alpha amino acid that I'm going to write here. C double bond O, and here is your OH. And then I'm going to write this CHH. This you can easily understand that this is your alpha amino acid, first one. And another alpha amino acid that I'm going to write is like this. Now this will be, again, this is glycine basically, 2 moles of glycine, double bond O, and OH is here. Now what I'm going to do is very simple. I'm going to remove this H2. Okay, if I'm going to remove this H2, what will be the product? It's very easy to understand. H2O will be removed from here. Wait a minute. H2O will be removed from here. And you can see what will form N, and here it is H, and again H, and then there is C. Let me take this as R. It will be for then it will be for general, or it will be, if you want to write it H, it can be, not a problem. So it will be like this: C double bond O, and then comes, wait a minute please, I just did it directly. Then comes C, and then there is H, then there is H, and then C double bond O, and then there will be, it will not be completed in this line. That's why I'm going to write it on the bottom. Wait a minute, please. So how we are going to write it, you can see this is your hydrogen, this is also your hydrogen, this is your carbon, this is your hydrogen, this is your hydrogen, and then comes C double bond O, and then comes nitrogen will be with one of the hydrogen because one hydrogen is disconnected, and then C H H, and then C double bond O, and this is your OH, and H2O is basically eliminated. Now as you can see, this part as you can see is basically your peptide bond, CO peptide bond, CONH. Is that clear, Sakina?
Yes, sir.
Okay, thank you. Please do write it.
Done, sir.
Very
Sir, 1 minute. I'm drawing the last structure.
okay. Let me know, Anushka, when you're done with this.
Done, sir.
Very good. Wait a minute, Anushka. Okay. Write a point with me, which is very easy. Peptides are further divided into di, tri, tetra, depending upon the number of alpha amino acid, depending upon the number of alpha amino acid combined. If it comes from two amino acid, it is dipeptide. If it comes from 3 amino acid, it will be tripeptide, and so on. Are you getting my point?
Yes, sir.
Please do write this.
Done, sir.
Very good. Now see how we are going to have this concept. You know, glycine, I already had written the structure of glycine. So this is your glycine, which is an alpha amino acid. And if you are going to combine with this NH and H with, wait a minute. So this is your alanine. This is your alanine. So what will happen? You can see this OH will be eliminated with H, so that H2O will be eliminated, and see what we will get. It's very easy. So we can easily write this part, and H. After this, there is 2 hydrogen with this carbon, and then comes C double bond O. OH is eliminated with this hydrogen, so C will be directly bonded with this nitrogen, and in the same way, this is your CH3, this is your H. So this will be your C double bond O, and then OH. Now this is your alanine, and this is your glycine, and together they are basically, since 2 alpha amino acids, so we can call it dipeptide, and we can call it GLY, glyala. This is how we write. Please do let me know when you're done with this. So to score in this chapter, you need to study all the NCERT concepts. Okay.
Done, sir.
Done, everyone. Just wait a minute, please. Classification, we are done with this. Peptide bond. Okay, let's start. The structure of this, what we call, just wait a minute, just look at the topic. Okay, nucleoside. Okay. We started protein after this. Just we are going to do the classification first. So right, classification of protein on the basis of shape. On the basis of shape. Please do write with me. On the basis of shape. So they are of two types basically, the fibrous and the globular. So we can write they are of two types. The first one is globular protein, and the second one is fibrous protein. So what is the difference between them? So we just divided protein in two parts on the basis of shape, and just write with me. So we can write they have nearly a spherical shape. They have nearly a spherical shape. And here we can write they have linear thread like a structure. They have linear thread like structure. These are soluble in water. These are insoluble in water. These are insoluble in water. Next is it has or it have alpha helix structure. And here it have beta plated structure. Next we can write example insulin albumin. And here we can write keratin, which is in hair, silk, wool. Its example. And myosin you can call it, which is present in muscles. So this is the type of protein on the basis of shape. Globular, which are spherical in shape. Fibrous, which are linear thread like a structure. Fibrous is insoluble in water and globular is soluble in water. Globular has alpha helix structure and fibrous is beta plated structure. And the example of globular protein is insulin albumin, and fibrous is keratin, which is present in hair, silk, wool, and myosin, which is present in muscles. So I think it's clear to both of you, right?
Yes, sir. Sir, I'm done writing.
Very good. Sakina and Alan.
Yes, sir.
Wait a minute, please. Sakina, are you done with this?
Yes, sir, done.
Very good. Now we are going to do the structure of protein. So right, the structure of protein. So the first one is basically primary structure. Primary structure. What is primary structure? So I'm going to define it right with me. It refers to sequence of amino acid in each polypeptide chain. In each polypeptide chain. And its structure is like this. You can see primary, for primary structure, the structure is like this. They are equal basically. I'm not able to. Wait a minute. So it's like this. This is your primary structure of protein. Now the second is basically your secondary structure, which is also very easy. Secondary structure, and it is very important. Let me define it for you. It refers to shape in which polypeptide chain exists, in which polypeptide chain exists, and it is of two types. Basically, it is of two types. The first one is alpha helix, and the second one is beta plated. Beta plated that we discussed there. I'm going to write the structure here. So please do write up to this. Let me know when you are done with this.
Done, sir.
Very good. Sakina, you are also done with this. Now see, how is the structure
Yes,
of this
sir.
alpha helix? Very good. And beta sheet. The structure is very simple. Helix is like this. And if you are going for sheet, it is like this. You got it? This is your beta sheet. Then comes your tertiary structure. And let me define it for you. It represents the overall folding of polypeptide. Overall folding of polypeptide chain. That is, that is further folding of 2-degree structure. Further folding of 2-degree structure. You know, this is we are going to talk about fibrous and globular. Fibrous, and the second is globular. So I'm going to show you how tertiary is. Tertiary is basically further folding of the secondary, and if you are going to further fold it, so how the structure will look like, it is basically like this. It will be like this is the tertiary. Then comes your quaternary, which is quite complex. Quaternary structure. And you can write it refers to a spatial arrangement of subunits with respect to each other. So basically you will look at like an NCERT, you will get the diagram of that. Okay. And then we are going to the next topic is denaturation, and then we will go nucleic acid.
Done, sir.
Yes, sir, done.
Very good. Wait a minute. Okay. Now, right to topic. Alan, you are also done with this.
Yes, sir.
Denaturation of protein. I think you have all studied this in biomolecules, biology. Yes or no? This is purely biology. So please do write the definition with me, then I'll explain. So everyone, it's very easy, as you can see. What is denaturation of protein? So whenever you are going to change any physical change or chemical change, like if you are going to change physical change, like you are going to increase the temperature or decrease the temperature, or you are going to do chemical change, like you are going to change the pH or anything else, the hydrogen bond that is present in protein will be disturbed, due to which globules which are folded will be unfolded or uncoiled and loses the protein which is now uncoiled due to the change in physical or chemical change, will lose its identity or biological activity. Basically, the protein will lose its biological activity, and this is called denaturation of protein. Is that clear? Like boiling of egg, and uh, when you prepare curd from milk, that is denaturation of protein. Please do write it.
Sir, done.
Very good, Alan. You are also done with this.
Yes, sir.
Very good, Sakina. What about you?
Yes, sir.
Very good. So, anyone have any doubts from in this?
No, sir, understood.
No. Now, next point regarding this, you can write during denaturation. Please write with me. During denaturation, 2-degree and 3-degree structure are destroyed. 2-degree and 3-degree structure are destroyed, but 1-degree structure remains intact. It will be as it is. As we can take an example of coagulation of egg white on boiling, or either you can take another example, that is curding of milk. Curding of, and I think it's easy, right? So please do write it. Then we are going to talk about nucleic acid.
Done, sir.
Everyone done with this? Alan, Sakina.
Yes, sir. Done.
Yes, sir.
Oh. Now write the last topic of this chapter, that is nucleic acid. So what we are going to do in on Wednesday? Amines.
Yes, sir.
So only amines left for the organic chemistry, right?
Yes, sir.
No. Is there any other chapter in organic or in second book of NCERT that is left? Or this is the only last chapter?
No, sir, only amine is left.
Okay. So these are basically polymers of nucleotides. These are polymers of nucleotides present in the nucleus. Present in the nucleus of the cell. These are also called polynucleotides. Polynucleotides. These are also called polynucleotides. These are of two types. The first one is deoxyribonucleic acid. Deoxyribonucleic acid, and the another is ribonucleic acid. Ribonucleic acid. So deoxyribonucleic acid, that is DNA, and ribonucleic acid, that is RNA. First of all, write it. Nucleic acid are of two types. It is present in the cell, and it is polymer of nucleotides. So it is also known as polynucleotides, deoxyribonucleic acid, ribonucleic acid. What is the composition that we are going to talk about? First write it, then let me know.
Done, sir.
Very good. Now see. Composition of nucleic acid. Composition of nucleic acids. So first one is basically pentose sugar. Pentose sugar. Then comes your phosphoric acid is also present. Phosphoric acid. Then nitrogenous base. Nitrogenous base. Then this is the three component that is present in nucleic acid. Is that clear?
Yes, sir.
But if you are going to be specific, like in DNA, sugar present is the name of sugar that is present. Sugar present is beta D 2 deoxy ribose that is present in DNA. And if you are going to look in RNA, sugar present is beta D ribose. So the question can be directly asked, which type of sugar present in DNA and which type of sugar present in RNA. It's quite easy. You just need to remember the name. Okay.
Yes, sir. Done.
All of you are done with this? Okay. Now see, the base that is present, it is also very important. Base present in nucleic acid or adenine, that is represented by A, guanine, that is represented by G, cytosine, that is represented by C, uracil, that is represented by U, and thymine, that is represented by T. So if you are looking in DNA, alanine, guanine, cytosine, and thymine, all these four are present. And if you are looking in RNA, if you're looking at RNA, so see, A, G, C, U is present. Alanine, guanine, cytosine, and uracil is present. Now look at this. nucleoside nucleoside, that means if there is nucleoside, that means sugar plus base both present. And if you have nucleotide that is in RNA, that is basically sugar plus base plus phosphoric acid. This is the difference between this phosphoric acid. So please do write up to this and then let.
Done, sir.
Very good. Now see, always remember the difference between this nucleoside, Indian, and nucleotide in RNA. Now, difference between this nucleotide and nucleoside. I'm going to distinguish between these two. Please do write with me. Oh, it is side. First, we wrote here side, nucleoside, and this is your nucleotide. So write the difference with me. Okay, it's very easy. Let me know when you are done with this.
Done, sir.
So it's very easy. As you can see, the difference in nucleotide and nucleoside. So this is the nucleoside. Basically is a compound formed by the union of nitrogen base and pentose sugar. Whereas in nucleotide, the union is nitrogen base and pentose sugar as well as phosphate or phosphoric acid that comes from phosphate. Next is basically it is component of nucleotide. It is component of nucleotide, and nucleotide is formed by the through phosphorylation of nucleoside. If you are going to react this nucleoside with phosphorus, and if there will be phosphorylation like acylation, alkylation, then nucleotide will form, and it is slightly basic in nature, the nucleoside, and this is slightly acidic in nature. Is that clear, both of you? Every one of you?
Yes, sir, clear.
Now, I'm going to differentiate between RNA and DNA. Alan, do you have any doubt? Sakina and Alan?
No, sir.
Okay, thank you. Please be regular in the next class, because in the next class we are going to start amines, which is a very important chapter. Difference between DNA and RNA. This is the last. So here you can write DNA, and here you can write RNA. It is double-stranded nucleic acid. And what about RNA is single? It is single-stranded nucleic acid. Next is it contains deoxyribose. It contains deoxyribose sugar. It contains deoxyribose sugar. It contains ribose sugar. Wait a minute. So please correct it. This is a ribose, deoxyribose, and this is only ribose. Next is it contains thymine, that means T, as a nitrogenous base. Nitrogenous base. Next, here you can write it contains uracil, that means U, instead of thymine. Next, we can write it is the genetic and hereditary material of the cell. Material of the cell. It is involved in synthesis of proteins. It is involved in synthesis of proteins. it is present in the nucleus of the cells. Nucleus of the cells, and it is present in both nucleus and cytoplasm. Cytoplasm. Please do write it and let me know if you have any doubt in it.
Done, sir.
Okay, everyone done with this? Anushka, Sakina.
Yes, sir.
Sakina, what about you?
Yes, sir, done.
Okay, so they here are complete. The chapter is completed. Biomolecules. In the next class that we are going to do is amines, and after this I would like to again ask that I forgot. So sorry. After amine, what is the which is the chapter we need to cover from the first book, Anushka?
Oh, sir, 1 second, I'll check and let you know.
I think solution it is.
Sir, solutions, DNF block.
And any other?
1 second, sir.
Okay, 3 chapters left. Okay, amines is a quite a small chapter. Then after amines, we will go for D and F block. Okay,
Yes,
is
sir.
that clear? So this is it for today. If you have any doubt, please do let me. Do let me.
Sir,
Yes.
so I think even chemical kinetics is remaining, right?
I don't know because I was not there.
Yes, sir, even that is remaining.
Chemical kinetics too.
Yes, sir.
So we must have extra class in order to cover this syllabus. This Sunday, this Friday, will be not free, but after this week, we can have extra class whenever you all are free together. Okay, in any
Yes,
day,
sir.
on any day.
Yes, sir.
Okay, so this is it for today. Thank you so much.
Thank you, sir. Bye, sir.